Yeah, like Aaron said, my thesis research is on the implications of Pacific Northwest climate patterns and the harmful algal blooms, and then, of course, the subsequent marine biotoxins. Next slide, there we go. So a little bit of background information. Phytoplankton, they're the most abundant living organism in the ocean. They are primary producers, meaning like they photosynthesize, and they provide over 45% of the Earth's oxygen, which I think is absolutely amazing. They're an important part of ocean's ecosystems and food webs, because again, they're primary producers, so they're like at the base of every food chain, and then you can have cascading effects from there. And shellfish consume phytoplankton. They're filter feeders and are dependent on phytoplankton. The one kind of negative thing along with phytoplankton, as great as they are and as many benefits they do bring to like the natural world, there's a thing known as harmful algal blooms. And basically what that is it's, a certain phytoplankton species, a dinoflagellate or a diatom. Typically it's a toxin that they release just naturally. It may be a metabolism byproduct, but if the phytoplankton is there and there are harmful algal bloom species, that toxin is typically there. And so then that can bioaccumulate in shellfish, because shellfish, again, filter feed and eat these phytoplankton, and that's what is known as shellfish toxicity. And that can really impact the shellfish industry and then, of course, the stakeholders within that. And here in Washington, it's like, it's like our culture. It's a big icon. People come to, Ocean Shores or Westport to clam. So it is a big deal. It's a $270 million industry, which I feel like in the grand scheme of things might not sound like a lot, but to like coastal and rural communities when there's not much other industry other than like, shellfish farming, it really can impact them. So yeah, moving on to specifically paralytic shellfish poisoning. This is the result of a biotoxin called saxitoxin that is released by a dinoflagellate called Alexandrium catenella, and it's in that image under microscope. So again, when they're thriving, when they're proliferating, if they're there in the water, saxitoxin is in the water, and so then subsequently shellfish accumulate it. And it impacts the shellfish in no way. It doesn't harm the shellfish. They're just bioaccumulating it, and it's just sitting there. But then when humans consume it, mammals consume it, birds consume it, etc., it has harmful impacts. It's actually a neurotoxin, so it can even be lethal to humans. In the symptom of like you can experience like muscle respiratory paralysis, and then like you asphyxiate if you don't have medical attention. I feel like it always sounds so doom and gloom when I describe that, but it's monitored and regulated by Department of Health. There's not people dying every year from this, but it's just an important thing to note that it is like a serious toxin that can hurt animals and people. So what influences harmful algal blooms like Alexandrium? Again, they're primary producers, they're photosynthesizers, so we would expect carbon dioxide and nutrients to impact them. And then also temperature can be a catalyst. And so that is what I've mostly focused on. So there's also this paper from Moore et al. From 2010. They looked at number of days over 13 degrees Celsius in Sequim, Washington, or number of days sea surface temperature is above 13 degrees Celsius in Sequim. And they looked at how that impacted shellfish toxicity. So in this figure on the y-axis on the left, that's shellfish toxicity, specifically saxitoxin concentration, which again is what causes paralytic shellfish poisoning. And it's micrograms per 100 grams. And I just want to mention also real quick that the action point, which is like what's deemed unsafe for human consumption, is 80 micrograms. And so that's just, so you have some sort of base to compare these numbers to. But yeah, so they found that Alexandrium does really well in warmer temperatures, 13 degrees to 17 degrees Celsius. And they found that number of days where sea surface temperature is greater than 13 degrees Celsius does significantly increase shellfish toxicity in butter clams. That's what's shown there on the figure. So what influences sea surface temperatures? So again, for this thesis research, I was looking at climate patterns, specifically El Nino, Southern Oscillation, and Pacific Decadal Oscillation. And I'll kind of go back and forth, but I will be referring to them as ENSO and PDO. So what is El Nino Southern Oscillation? Firstly, as you can see this figure here, it's the ENSO index graphed over time, thanks to NOAA. That y-axis is the degrees Celsius. And it's the climate pattern of like sea surface temperatures and atmospheric pressure fluctuations. And so basically what's going on, real quick, is just describe how like the mechanisms of it. You have the eastern trade winds, moving from the east to the west along the equator. And so when you have strong eastern trade winds, it's pushing water westward. And when you have water, being pushed away from, the continent, you then have more upwelling. And so when you have strong eastern trade winds, you have more upwelling and more cold water coming up. And so that would be a La Nina phase. And then El Nino would be the opposite of that. You have weak eastern trade winds, less upwelling. Then you have like warm, stagnant water. And so that would be an El Nino. So warm phase, El Nino, cold phase, La Nina. And then moving on to Pacific Decadal Oscillation. Again, this figure here is the PDO index graphed over time. Again, thanks to NOAA. And the PDO, it's an ENSO-like pattern of climate variability, and it's describes a lot of our sea surface temperature variability and anomalies here in the Pacific Northwest. And again, there's warm and cool phases. As you can see here, obviously the red lines are warm phases and the blue lines are cool phases. And so the main drivers of Pacific Decadal Oscillation are ENSO via the mechanisms I just described, and then the Aleutian Low, which is basically, it's like a semi-permanent pressure system that impacts like weather and climate, and then also subsequently sea surface temperatures here. And then one last thing I want to mention about PDO is, by the name, you would guess that it phases can last decades, but as more recently, the phases have the frequency has increased, so it's going back and forth between phases more frequently than before. So my research question: to what extent do warm and cold phases of PDO and ENSO correlate with saxitoxin concentration? Prairie Lake shellfish poisoning here in Washington. Also, I kind of jump between like saxitoxin concentration, shellfish toxicity, PSP, and those are all kind of like interchangeable. And so I just wanted to mention that if anyone gets confused by that. So moving on to my methods, the location for all this data is from Sequim, Washington. I also wanted to mention Moore et al. They laid down like all of the framework for me to be able to do this research. But so on the left there, that's a GIS map I created working for Department of Health for all of our biotoxin sample sites, just to have reference for newer people trying to find the sites. You can go to the GIS map, click on it, and it gives you, all the information you need. And those are the two current sites. And then on the right there, that's a map from Moore et al. Showing current and past biotoxin sample sites. And if you don't know where Sequim, Washington is, if you just go up north of 101, it's northern Washington. It's a gorgeous area, and that is where all the biotoxin and sea surface temperature data is from. It's from up there. So then my data, again, as I mentioned, the ENSO and the PDO index data, that is all thanks to NOAA. And then sea surface temperature data, that is thanks to DFO, or Department of Fisheries and Oceans. They're up in Canada. And then the biotoxin data, that is thanks to the Washington State Department of Health. So moving on to my statistical analyses, I used Excel to manage so much data. I felt like so much of my time and effort was just trying to clean all the data. And then, I ran my tests in RStudio. And so I used simple linear regression and Pearson's correlation, to look at the relationships with sea surface temperature, PSP index, PDO index, and then ENSO index. And the timeline is from 1957 to 2022. The Moore et al. Paper that laid down a lot of this framework is up to 2007. And in their paper, they actually did not find any significant findings between these variables. So starting on the left, that's mean annual temperature graphed over time. I ran a linear regression on that and found significance. So it seems that temperature is increasing over time. I think not too surprising to most people. And I do want to emphasize that I use mean annual temperature, and that's why the temperatures seem lower compared to the Moore et al. Because you'll see that I have no temperatures over 13 degrees Celsius, whereas the Moore et al. Looked at temperatures over 13 degrees Celsius. And then this is on the right is PSP over time, highly variable. There was no significance. And then looking at PSP and temperature, ran correlation. You can see they're positively correlated, but pretty weakly, so not significant. Again, I use mean annual temperature to try to represent how temperature is increasing over time, but I think the number of days over 13 degrees Celsius might represent the relationship better as more used. And then here is looking at PDO index and SST. So on the left figure, you have Sequim, sea surface temperatures, and then PDO index. And I think this really shows how well or how strongly PDO and also ENSO influence Sequim, sea surface temperatures. You can see how closely they follow each other. So significant, strongly correlated. And then moving on to shellfish toxicity. So on the left here, we have PDO index on the x-axis, and then PSP toxins or saxitoxin concentration on the y-axis. And there is a significant positive relationship between PDO index and PSP, as you can see there from the p-value. And then on the right, similar findings between the ENSO index and PSP concentration. We have a significant and strong positive correlation, which is exciting for me. So a little bit of discussion. So we've seen the sea surface temperatures. It seems that they're continuing to rise over time. Part of this could be to the ENSO and PDO influence on sea surface temperatures. Moore et al. Showed that temperatures over 13 degrees Celsius, so warmer temperatures, lead to more Alexandrium, thus more shellfish toxicity. And one thing of note that I wanted to mention is, if y'all remember, from 2013 to 2015, there was that big marine heat wave called the blob. That led to, on average, much warmer sea surface temperatures, and that also could have led to, a great opportunity and window for Alexandrium and PSP. And then in conclusion, kind of just like to wrap it all up, sea surface temperatures are increasing, partly due to ENSO and PDO. So that's increasing Alexandrium, and that means that it is increasing parallel shellfish poisoning. So I hope that this thesis research can contribute to hopefully better monitor and regulate shellfish toxicity through the Department of Health. That is all. Are there any questions?